doing what we're doing. So firstly, I'm Tracy Calvo-Joshua. I'm the scientific lead at PHA4GE. In the background, you may not see him, is the host, Jamie Southgate, who is our operations lead. And he actually was previously one of the co-chairs of one of the working groups at PHA4GE. So if you're not familiar with PHA4GE, PHA4GE stands for the Public Health Alliance for Genomic Epidemiology. And at PHA4GE, we've got several working groups happening, and we've also got projects happening at the moment, all concurrently. Where you are sitting at the moment is the pipelines. I'm going to see if I can turn this on. Laser pointer. You are sitting here at the bioinformatics pipelines and visualization group, which is obviously quite long, so we'll be calling it PipeViz from now onwards. And this group is predominantly focused on developing guidance documents for various types of bioinformatics or doing bioinformatics and genomic analysis on various pathogens and even applications such as wastewater surveillance. And just to let you know, we've got the bigger overarching PipeViz working group, which is led by Emily Smith and Grotigan, I'm probably not saying his name properly, but these co-chairs and obviously those going ahead of them have done a fantastic job in developing a whole bunch of guidance documents already. As you can see, some of them are listed there: SARS-CoV-2, Mpox, HIV, influenza, and some of them are ongoing, such as the foodborne pathogens sub-working group. And speaking of sub-working group, so this is where the phylogenetics comes in. As they've started developing these guidance documents, they realized that there was so much that needed to be said about phylogenetics that there's actually enough information for this to become a guidance document on its own. And so the phylogenetics sub-working group was kind of soft launch. We've got the co-leads of that sub-working group in place, who is Dr. Shavari Narendra. I don't think she'll be able to join us today. She's on maternity leave. But Rita Mikari Chowki is here, and she's also one of the co-chairs of this sub-working group. And so just to let you know, you are welcome to, Jamie, if you could just put the link for them to register. You're welcome to register to the PipeViz working group. Once you join, you will receive the necessary documentation in order to then join the next meeting where you will get the necessary information to join the sub-working group. And a reminder to you, these guys are as lovely as their pictures look. You will feel right at home. So please do not think you need to be an expert to join. You're welcome if you're a student. This is a community-driven project, just as all of the others are. So we definitely need your input, such as, we think this case study should be useful, or I actually don't think this is useful for my community. There's so many ways in which you can contribute as somebody that understands your local community. So please feel free to join. And also recently we've taken a decision to actually change our dynamic documents, which are here on GitHub, into manuscripts. So for those of you who want to join, there's obviously an opportunity for authorships on manuscripts, so please do join. And then lastly, house rules. Not a lot, just 3. We don't like rules. The first is just be respectful to one another in the comment section. That being, or in the chat. The chats are for you to introduce yourself, to chat to one another, but please do not post your questions in there. There should be a Q&A, although Jamie, I cannot see the Q&A, but there should be a Q&A. Am I just missing it?
It's at the bottom of the screen. I can see it.
Oh, I can't see it because I'm in presentation mode. But there should be a Q&A. Please use that box for your questions. And then lastly, as you can see, the session is being recorded. So if for any reason you have connection issues and you're not able to watch this entire webinar, then please feel free to actually go to our YouTube. And I've specifically written the PHA4GE with the 4 in red so that you remember that there is a 4 in PHA4GE in case you find bacteriophages when you look for PHA4GE. That being said, I'm going to just stop sharing, and I want to introduce Dr. Zoe Dyson right now. who is an associate professor, something's going on here, at the London School of Hygiene and Tropical Medicine. And she has actually joined us before. If you've missed it, again, you should be able to find it on our YouTube channel. And just to give you some context again, or she has done an introduction, phylogenetics, but this one specifically is on leveraging phylodynamics to understand the epidemiology of enteric fever pathogens. Thank you, Dr. Zoe, for joining us again. I'm going to hand over to you. If you could share your screen, we're looking forward to what you have to share.
Thanks, Tracy, for the invitation to speak and the very kind introduction. I'll get my screen sharing set up. Okay, could I just confirm that you're seeing slides and not presenter view?
Seeing the slides and can hear you clearly.
Wonderful, thank you so much. So thank you everyone for joining today. It's my pleasure to speak about how we can leverage phylodynamics to understand the emergence and spread of not just enteric fever, but bacterial pathogens generally. I'm assistant professor at the London School of Hygiene and Tropical Medicine, and most of the work that I do here is computationally based and concerns better understanding the transmission dynamics and evolution of bacterial pathogens. So today's webinar tutorial is going to cover a couple of different topics. Firstly, we're going to talk about some key concepts around phylodynamics. We're going to walk through an overview of the steps that are involved in a phylodynamic analysis and what each component of these means. The third section here I've just called Dated Trees Plus because there's still quite a lot of really cool analysis you can do to better understand the epidemiology of bacterial pathogens once you have your data tree. And I just wanted to show you some of the different software tools and options that are available to you as an analyst. And I wanted to finish up with a case study where we look at the application of phylodynamic methods to understand the emergence and spread of multi-drug resistant typhoid in sub-Saharan Africa. So firstly, let's talk about what phylodynamics is. So in the previous webinar I gave in March, I gave an overview of phylogenomics. And at that point, we were talking about sort of the concept of this being an area of study where we could look at the genetic relationships between different biological entities, be those viruses, be those bacteria, fungi, all sorts of different organisms and biological entities. And I went through then that this was not a new concept. This was a fairly old concept, first attributed to Charles Darwin in around 1837, where you can see that he sketched out this sort of idea of what a phylogenetic or a phylogenomic tree could look like and how it would explain different evolutionary relationships. In the whole genome sequencing era, we tend to use nucleotide or amino acid sequences in a field known as molecular phylogenetics to infer evolutionary relationships. But previous to this, it was common to use other trait data of organisms, for example, bird beak shapes or other traits. And the focus of this webinar will be molecular phylogenetics in the context of using whole genome sequencing data, core genome alignments of nucleotides that have been derived from bacterial pathogens. So that brings me to, well, what is phylodynamics and how is this different to phylogenomics? And phylodynamics is really a relatively new term that was coined in around 2004 by Grenfell and colleagues. And this was described at that point as the melding of immunodynamics, epidemiology, and evolutionary biology. And the reason we might want to bring together these different concepts and different disciplines is that by bringing all of this information and knowledge and techniques together, we can better understand the evolution of antimicrobial resistance and virulence, the sort of geographic and temporal scales on which this can emerge. And this can help influence vaccine design and coverage, and it can help us understand the emergence of new public health threats. So phylodynamics typically revolves around inferring a dated phylogenetic tree and using that tree to sort of model various aspects of the pathogen population that you want to better understand. So phylodynamic methods were initially developed around viruses, I believe originally RNA viruses. And for this webinar, we're focusing on how those tools have been adapted and applied to bacterial pathogens. And while there will be a bit of overlap on how you'd approach this for indeed other pathogens, the focus really here is on the processes you would go through for specifically for bacterial pathogens. So last time I was lucky enough to present to you all, I went through sort of a diagram that looked very much like this on how to read and interpret data from a phylogenetic tree. And for this webinar, I have created a slightly different version of that figure where I have replaced the scale bar with time, as you would expect to see in a dated phylodynamic tree that you've inferred. So I've sort of grayed out the other points on how you would read and interpret a phylogenetic or a phylogenomic tree. And this time I've sort of just highlighted one key difference that you will notice is that when you're looking at a phylodynamic tree that's been inferred using usually a Bayesian approach, the branch lengths that you're looking at represent time. So if I would be thinking about better understanding the evolution of, say, sequence A and B, these two tips in our phylogenetic tree, and looking at their ancestor X, I could see that the divergence date for X is probably around 1975, and that's when that hypothetical ancestor that existed in the past that we don't know the sequence of is likely to have emerged from the data that we have in front of us. So I've just expanded on these thoughts a little bit here as to what you would be looking at when you're looking at a dated phylogeny. And as I said, most of the interpretation is the same, but when we start looking at, say, our root node Z here that represents the most recent common ancestor of all other nodes, internal or internal, ancestral or contemporaneous, in our data set, when we're looking at, again, the branching and the position of the root node, we can see that the root date, also known as a divergence date of Z, or the time to most recent common ancestor of Z, is sort of the date of that we're sort of seeing is probably a little bit before 1925. And depending on the data that you're analyzing, for example, if you were analyzing whole genome sequencing data all from one country of one particular pathogen, you could interpret the date of Z as the emergence of that pathogen in a particular country. So it's a really contextual to what you're actually analyzing when you're inferring these particular trees. And I'll touch on this again later, but all of these estimates of divergence dates are accompanied by what is referred to as 95% highest posterity densities. These are analogous to 95% confidence intervals, and you might see that become slightly wider at the root of the tree because the model is usually doing the most inference back in time at that point. So something else to think about is how we might use this other than looking at when a pathogen's potentially emerged in a location would be looking at the evolution of traits such as antimicrobial resistance. So looking at this slightly modified figure, if we could see evidence of antimicrobial resistance in samples C, D, and E, we could then look to their shared common ancestor Y, and the divergence date of this is around the 1950s. So we could assume from the data presented here that that resistance has emerged in that cluster in around 1950. So this is some of the sort of observations you can make when you start looking at dated phylogenetic trees. So some of the key concepts that are helpful to know about when you start thinking about interpreting or indeed analyzing data in this way is that when we're looking at genomic epidemiology of bacterial pathogens, we're making the assumption that our epidemiological processes are occurring on a similar timescale to genomic change. And we have this important concept of a molecular clock, and this is the concept that pathogen evolution is occurring at a roughly consistent and measurable rate over time that we can compare to genetic change and use this information together to infer the dates of past events. So when we come to calibrating our molecular clock, and we'll talk about this later on, we're usually calibrating that data when it comes to a pathogen example using the sampling dates or the tip dates for the samples or the sequences you wish to analyze. And this can come at a variety of different resolutions. This could be the year of isolation of the pathogen, it could be the year and the month, or it could be day, month, year. And it sort of depends on your purposes and the data available to you as to what scale of resolution you're using for your dates. So we're calibrating our molecular clock using our sampling dates, and genomic change is measured from your core whole genome sequence alignment of the pathogen under study. And this is all done by software for you. So another really important concept I'd like to introduce at this point is that of temporal signal. And this is referring to having a sufficient accumulation of genetic change occurring between different sampling times in your population under study that you can recognize this relationship between genetic divergence and time. i.e. that the pathogen is measurably evolving and you can indeed calibrate your molecular clock and produce a sensible analysis. So when we have that temporal signal present, and I'll talk about different ways of establishing that later on, you can go ahead and infer your dated tree where your branching times may reflect transmission dynamics. So now that we have an idea of what phylodynamics is, the key plots involved in this and the key data points and some of the key concepts, I'd now like to introduce a brief overview of phylodynamic analyses. So when you type into Google phylodynamic tree inference software, you'll see quite a lot of different tools come up. I've just included on this slide some of the more popular tools. What I'd like to talk mainly about today is the tools that I'm going to show you on this slide, and there's 4 of them. These are some of the most popular tools for carrying out these types of inferences that are available today and that are most popular today. We have Beast 1 and Beast 2. I think it's important to point out that Beast 2 doesn't supersede Beast 1. These are simply sort of a diverging point, if you will, in the software where different groups among the community wanted to develop different aspects of the tool, and they made the decision to split that into 2 different versions. The choice as to whether you use Beast 1 or Beast 2 very much for me depends on the types of models that I want to use and the type of analysis that I want to do and what I want to extract from that. And I move between those 2 tools depending on what I want to do. The other 2 tools I would like to point out to you are both R packages. These are tree data and backdating. Beast is a wonderful tool, Beast 1 and Beast 2. They have a lot of different prior distributions you can set, and a lot of different priors that can be set. They're really easily extendable. There's a lot of different options and different models you can use in those. But if you have thousands of samples, indeed maybe over 300 to 500 samples, you may find that you're exhausting your computational resources and that the analysis is becoming computationally intractable. The reason I'm highlighting these R packages is that these are actually able to process data in a more efficient way using just a maximum likelihood phylodynamic tree, such that you can process large volumes of data, but you do lose some of the extendability in the types of models you can use and access with these tools. So again, this is another consideration of which tool you might want to use. If you're working with lots and lots of samples, the R packages may be more appropriate to you depending on the analysis you want to do. But they're all excellent tools, and I highly recommend all of these. So unfortunately, I can't background Bayesian statistics as much as I would like. You could indeed run an entire year-long course on this and still have more to learn. But I think it is worth pointing out that many of the tools I just showed you, but not all, do in fact use Bayesian statistical frameworks to carry out dated tree inference. And some of the key concepts and outputs and inputs that you will notice if you're not used to working in a Bayesian framework will be that every model parameter that you want to use and you want to calibrate and you want to parameterize will have a prior distribution of values that are supplied to it. And these reflect our assumptions that we know about our data set before we've done any analysis. Similarly, when you are extracting your results from the analysis, you will also receive the posterior distribution that contains your results as another distribution. And this contains a distribution of trees. It contains a distribution of, say, substitution rate estimates and divergent states. And because you're working with a distribution, that's why you have this precision estimate that comes to you via a 95% higher posterity density that you could think of as being sort of analogous to 95% confidence intervals under a frequentist statistical framework. And that posterior distribution is sampled and provided to you via a Markov chain Monte Carlo sampling technique that explores the posterior distribution, which kind of you can imagine as a 3D landscape of peaks and troughs that this system is set up to explore and provide the data to you in a series of different steps as the model explores this space that contains the end results of your analysis. So given that, I'd now like to overview the key steps in doing a dated analysis. Depending on the tool you're using, not all of these steps may apply, but generally speaking, most of them are used in most dated tree inference software tools. So the weird bit to start off with is probably your first step is actually inferring a maximum likelihood phylogenetic tree. And I believe Con and Mian backgrounded this in May. As one of the options you might select in terms of doing a phylogenetic analysis. So you want your maximum likelihood phylogenetic tree at step 1. This is something you might infer after refiltering for recombination with gubbins and then using RAxML to get your maximum likelihood tree. And you also want your tip dates or your sampling dates. So these might be your isolation date or your year of isolation. The next step is to carry out a root to tip regression analysis that looks at genetic divergence in terms of the path of the sample from root to tip for every sample you're analyzing, and then it carries out a regression analysis with that information and the sampling dates that you've provided. And depending on how that looks, you might decide that you have enough potential for a weak or a moderate or a strong temporal signal in the data set that you're looking at, that you then might want to carry on and do a formal dating analysis. So the next step of that is usually model testing, where you will compare several different models to see which best fit your data. And if you're getting good results at that stage, you might proceed to trying to run your formal analysis and attempt to get your best fitting model to converge. And at that point, if that's all happening and that's all going well, you would want to confirm that you have temporal signal in your data. And that's usually carried out through something like a date randomization test or BETS analysis. And we'll talk more about each of these concepts along the way. Finally, if you can confirm that you do have a robust temporal signal in your data, you can go ahead and infer your dated tree, which is often referred to as a maximum clade credibility tree. So let's take a look at this root to tip regression analysis first. So Tempest is a really commonly used software tool for carrying out root to tip regression analyses. And what this requires is a recombination filtered maximum likelihood phylogenetic tree and your sampling dates. So these are your dates of isolation in most cases. And what these are going to produce for you is a linear regression of your data. And I've just included several different analyses carried out by a really wonderful paper by Sebastian Duchenne, published in Microbial Genomics in 2016, where they explored temporal signal and a variety of other factors in a variety of different bacterial populations. And you can see that some of these have quite strong temporal signal, some of these have quite weak temporal signal. So different pathogens and different data sets will all look a little bit different each time you do an analysis. And if you are used to looking at viral data, these will look a little bit noisier to you. But this is sort of a part of looking at bacterial genomic data with these techniques. So with this, you're looking that you can calibrate your molecular clock with sampling dates or tip dates, and you're getting reasonable results that do suggest your data have clock-like behavior and that it's worth progressing your analysis to the next stage. So after you've gotten a good result from your root to tip regression analysis that suggests that there may be temporal signal in your data, you're going to want to progress to your model testing stage. So the type of input data you might need at this stage of your analysis will vary a little bit, but it could include a recombination filtered core genome SNP alignment. It could include a recombination filtered maximum likelihood phylogenetic tree. If you're using some of those R packages I highlighted, that's probably the case for those. It would require your sampling dates, as we've mentioned previously, a substitution model. Probably for most bacterial pathogens, this would be some flavor of a GTR, generalized time reversible substitution model that captures the transitions between different nucleotides that you expect to see in your data and the rates that you would expect those to occur at. It would include a clock model. This is usually a strict or relaxed model, and tree priors that you might also have heard referred to as demographic models that attempt to capture changes in the population size over time. So if you're doing this analysis in either BEAST or BEAST2, you would be parameterizing the model using Beauty. This is a graphical user interface tool, much like Tempest is also, I should mention. But if you're using the R packages, these will have specific functions to do this. So usually you'll be testing multiple clock models in combination with multiple tree priors to determine which best fit your data. And there's a couple of different approaches that people use for this. Some people will use an informal approach to model testing and determining which model best fits the data, or which model combination, best fits the data. This can be the examination of skyline plots to look at changes in the effective population size over time, which we'll talk about later. Or examining the posterior distribution or the results that you have for your rate coefficient of variation variable as well. There are also formal methods that examine the marginal likelihood of your different models and the posterior distributions. These are called path sampling and stepping stone analyses, and from those you can actually calculate Bayes factors that will help you decide which model best fits your data. or indeed which model combination. No matter which of these approaches you're using, you want to make sure that when you do your model testing and you're trying to select that best fitting model, that the effective sample size, this is the number of independent draws from that posterior distribution that you're getting from that Markov chain Monte Carlo model, is high. What high looks like depends on a little bit, one, the analysis you're doing, and two, the software tool that you're using. But generally, we're talking anywhere from about 200 to 500 for the parameters that you're interested in estimating using this technique. So when I'm talking about model testing, I'm talking about testing different clock models and different tree priors that you might sometimes hear referred to as demographic models in combination together. So the set of models I'm going to sort of show you here might not be suitable to all data sets, but these are really commonly used in the field. So in terms of your clock model, that could be a strict clock or a relaxed clock, which you might see sometimes referred to as an uncorrelated clock. Under your strict clock model, the assumption is that every branch in a phylogenetic tree evolves according to the same evolutionary rate. Whereas with your relaxed clock, it's a little bit more flexible. It allows every branch of a phylogenetic tree to have its own different evolutionary rate. You're then going to want to test each of those clock models with different tree priors. And the two that I've demonstrated here are the constant coalescent model and the coalescent Bayesian skyline model. So with your constant coalescent population tree prior, this is assuming that the effective population size or the population size has remained constant throughout time. Your coalescent Bayesian skyline tree prior allows for more complex dynamics. This could be population size increases or decreases that have occurred over time, evidence of bottlenecks and such. So in the end, if you were using this approach, that might be appropriate for some data, but not others. The combinations that you'll be testing is your strict clock model with your constant coalescent tree prior, your strict clock model with your coalescent Bayesian skyline model, your relaxed clock model with your constant coalescent tree prior, your relaxed clock model with your coalescent Bayesian skyline tree prior, and you're going to compare the posterior distributions, either an informal or a formal model testing approach to determine which best fit your data. And if you haven't come across this sort of concept of a coalescent before, it's simply that, as you can see in this diagram here, that we're looking at our samples, we're assuming that these coalesce at some point in the past to a common ancestor, and at that point, the evolution and the ancestry is shared between these samples, so it's working backward in time. So we found our best fitting model, we're happy with that. We now want to progress to doing our analysis and hoping that that will converge and we can get some good results and eventually infer a dated phylodynamic tree. So if you're using the BEAST approach, you would be looking at this using setting up and running your models are set up in Beauty, as I said before. You would then be running your analysis in BEAST or BEAST2, depending on which version you're using. Or if you're using one of the R packages like backdating, there would be a function that would allow you to specify all of this. And at the end, you want to look at your posterior distribution to see if you're getting convergence. And if you're using the BEAST or BEAST2 tools, this would be a tool called Tracer that has a graphical user interface that you can see here. If you're using the R packages, many of these leverage another R package called Coda that can help visualize traces, but also report on your effective sample size, remembering that we want that number to be high. We want to know that we've well sampled our results or our posterior distribution. So when we have convergence, we have good mixing, meaning that the MCMC has explored the probability space, that 3D landscape I was talking about earlier well, and that we're getting good results out of this. So when that happens, it should look like this larger image that is often referred to as a hairy caterpillar, and that's indicative of that good mixing. If it's not well mixed, it might look something more like the smaller picture that you can see here, in which case you might choose to do more independent runs and combine those. That's something that you can do until you achieve convergence, but it may also be that your data and your priors won't actually ever converge in some cases, and that would be a stopping point where this analysis probably isn't appropriate to the data set that you're working with. Another thing you might want to know is that the first portion of your trace or your posterior distribution that you're looking at here is often removed. It will take a bit of time for that model to stabilize and get to the point where it's a hairy caterpillar. And this usually involves removing a portion of that trace known as burn-in. And that usually is at least 10% of the run. And that's removed from any estimates that you want to make from this, such as your substitution rates, your divergence dates, or indeed when you come to put together your final maximum clade credibility tree. As I mentioned before, your ESS values, and you can see those displayed in the larger image here in this panel. It's reporting all these numbers for you. That should be high. And as I mentioned, depending on the tool and the analysis, what high means does vary a little bit. So one other important thing you want to do at this stage is you want to set up another run using your best fitting model, and you want to select an option that is called sampling from priors. And this is essentially ignoring all the data you fed to the model and running the model without data to confirm that when you look at your results and compare it to the model you've run with the data, that your priors aren't being overly influential and that they're appropriate to the data. So you're making sure that those priors aren't being overly influential on the results that you're getting. And you can do that by comparing distributions in Tracer. So we've inferred our ML tree, we've done our root to tip regression, we've done our model testing, and we're getting convergence. Yay. The next step is to confirm that we have temporal signal. So as I mentioned earlier, there are indeed a couple of different approaches you can use for this. A sort of more formal approach that was published in 2020 by Duchenne and colleagues is this Bayesian evaluation of temporal signal, or BETS analysis. What this does is it does a formal statistical analysis comparing model fit of your model with the correct sampling times provided. And then it also creates a separate model and compares to that model where you've made all of the sampling dates the same. And it looks at which model fit is better. And if the model fit with the actual dates is better, then you have some temporal signal in that data. There's a lot more to that approach, but I would encourage you, if you're interested in using this, in taking a look at this excellent paper by Duchenne and colleagues published in 2020. Another approach that's quite commonly used is a date randomization test. So this is where you take your run with your best fitting model, and then you rerun it 10 to 20 times. And each time you're rerunning it, you're scrambling the dates, randomizing them, if you will, different ways. So all of the actual sampling times are assigned to different tips in the tree randomly 10 to 20 times, depending on how many times you're running this. You're rerunning your analysis with Beast or Beast 2. And then in Tracer, you're looking at the substitution rate that you're getting from that model. And what you're looking to see is if the black bars that you can see here in this box and whisker plot, if those black estimates there representing the actual dates of the sample are not overlapping with the randomized values. And this is giving you an idea of what the expectation of the rate estimates for your model and prior combinations would be in the absence of temporal signal. So you're hoping that either the mean estimate is elevated compared to those from the randomized data, or that your 95% HPDs, those are like a little bit like confidence intervals we talked about under a Bayesian framework, again, are separated to the rates that you're getting, the substitution rates that you're getting over time to the actual values. And again, I've provided some examples of that from another paper from Duchenne and colleagues in 2016. This is the same one I showed you for the regression analysis, so I do recommend taking a look at that. And if you want guidelines on interpreting these, there are some criteria in that paper as well. So we've done our root to tip regression, we've found our best fitting model, we're getting convergence, and we've been able to confirm that we do have a robust temporal signal in the data. The next stage is to infer our maximum clade credibility tree. And this is the final tree that you would use in your publications, your reporting, that you would use with other tools to do further analysis. And it's where you're extracting your timelines for important evolutionary events, such as the acquisition of resistance or virulence or other traits like these. These are our divergent states would come from this particular trait. So as I mentioned before, under our Bayesian framework, we're dealing with posterior distributions, and a bit unlike your neighbor joining approach, for example, instead of dealing with one tree, you now have a distribution of hundreds, if not thousands of trees. So if you want to look at all of those trees together in a sort of truly Bayesian way, what you can do is you can view all of these trees and how they overlap and where the most confidence is among the topology for these using a tool called DensiTree. And I'm just providing an example here of 3,000 trees that have been overlapped in this particular tool and visualized in this way. As I mentioned earlier, you can see that your 95% higher posterity densities do get a bit wider at the root here because the software is doing the most inference back in time at that point, and you start to lose a little bit of precision in a lot of cases. So how you get to your one tree to rule them all out of this analysis that you can present in your publications and do further analysis on is using a tool called Tree Annotator if you're working with Beast and Beast 2. And there will be ways to extract this object in R if you're working, for example, with backdating. Now, in the context of R, this maximum clade credibility tree is a tree that is within the inferred distribution of trees that has the highest product of posterior clade probability. So it has the most credible topology out of all of the trees that are contained within this distribution. And from this, the software is going to annotate on traits of interest. This is usually your divergence dates that have been calculated when in this dating process, as well as their 95% HPDs around them as well. So you can see when we bring this down to our MCC tree in the bottom portion of this plot that we now have these sort of, I guess, error bars on here showing you the 95% HPDs and how much precision we have on our estimates of dating at those particular ancestral nodes in the tree. And if it's of interest to anyone, that was plotted in ggtree, which I find to be my favorite way of plotting beast trees. It's an R package. It has a lot of flexibility, not only in being able to plot things like error bars onto your trees, it also has flexibility to plot AMR and other data next to it in the form of a heat map and to annotate different nodes in different ways. So I highly recommend that as well. So we have our dated phylodynamic tree. We've looked at some dates of interest. What else could we do with this if we wanted to sort of take our analysis a bit further? So one of the things that I think is really great to be aware of, particularly if you're a modeler, is that when you look at Beast 2, for example, I've just provided an overview of the package manager here. So these are different additional packages that are accessible through Beast 2, for example. And for example, the one I just highlighted here, EpiInf, allows you to attach a compartmental model to better understand epidemic dynamics, where you could look at, say, individuals moving between being sensitive, infected, and recovered or removed using this approach if you wanted to do some extra inference to better understand maybe an outbreak or something like this. Another thing that's quite helpful that I alluded to earlier is looking at changes in the effective population size of your pathogen population over time. And I've just highlighted an example here from Ingle and colleagues that was published in 2019 in Nature Communications. And this is an example of the decline and expansion of different sublineages of multi-drug resistant monophasic typhimurium that causes gastrointestinal diseases. And this was an example of lineages circulating in Australia. So this concept of effective population size is the idea of you're modeling an idealized population that shows the same rate of change in genetic diversity over time as the population that you're studying. But the thing to be aware of if you do want to pursue this analysis is that these plots are really sensitive to sampling. So if you're not certain that you have representative sampling in the data that you've collected, this may not be appropriate to the data you're working with. So as you can see in the example from Ingle and colleagues, we can see that lineage 1 has declined over time in the most recent years, whereas lineage 2 and indeed lineage 3, we're seeing more expansion of those particular lineages over time. And if you wanted to carry out this type of analysis, you could use an R package called SkyGrowth to do so. It's excellent. I highly recommend it to you. The other thing you can do with SkyGrowth is you can use this to infer the basic reproduction number of your pathogen population over time. I think most of us are probably all too familiar with R0 after the COVID pandemic, but if you need a fresher, this is a measure of how contagious a different pathogen might be, and indeed how many secondary cases might be derived from a case in a sensitive population. So you can model this from your dated phylodynamic tree, and you can provide additional information such as the infectious period that allows you to do this. And you might want to do this to look at changes in R0 over time or different R0 values for different lineages of the pathogen population. So another thing that you can do once you have a dated phylodynamic tree, if you're working with a rapidly evolving population, might be to try to reconstruct a transmission chain. There's a lot of conversation over what constitutes a rapidly evolving population. Indeed, many bacterial pathogens can't be considered as this. And if you're looking at sort of finding out more about that, I do recommend to you this study by Campbell and colleagues published in 2018 in PLOS Computational Biology. So one way to do this is to infer a transmission tree, and one way you can do this is with the TransPhylo-R package. And this allows you to look at changes in host while accounting for within host genetic diversity over time. So you can see in the dated phylogenetic dynamic tree down the bottom here, we have different colors representing transmission between different hosts in the population. There's a more diagrammatical version of that transmission tree up the top here. So this might be useful to reconstruct transmission chains of rapidly evolving populations. Another thing that's quite useful to do, and I'll show an example of this in a case study later in this presentation, is you might want to reconstruct different traits of your pathogen population. So you can do this either using discrete or continuous traits. With discrete trait mapping, essentially what happens is that you're mapping different states to the tips of your phylogenetic tree and then reconstructing back in time what the traits would have been at the ancestral nodes and looking at rates of change between these. So if the trait can be described using an evolutionary model, this would allow estimation of its rate of change along an evolution along the phylogenetic tree. So when you're using geography as your discrete trait that you're mapping, or even your continuous trait, this is often termed phylogeography. And this can either be using discrete traits, like in the example here, such as city or country or region, or it could be using a continuous trait like GPS coordinates. What you can also do to extend that approach a little bit is capture what are referred to as Markov jumps and rewards. And this is sort of an explicit way of looking at how many transitions you see between different states or indeed different geographical locations along phylogenetic branches in your tree. So again, I'm pointing towards this example from Ingle et al. From 2019, where they actually looked at the sort of geographic state changes using this method in their analysis. And what they were able to find is that there are a lot of importation events into Oceania, where indeed Australia is located, from Southeast Asia and Northwest Europe. So it can be useful in quantifying these events. Another way we've attempted to use this in the past was to model age-based transmission of typhoid. And we've represented it in a slightly different way, but we looked at how many transitions we were seeing along our phylogenetic tree across different patient age groups. And what we could see was that in this case, the majority of these transmissions were occurring in this 5 to 15-year age group. And we also looked at Markov rewards, which is the time that the model has been spending in these different states along the way. And you could sort of consider this as the time it's been circulating in that patient age group. And again, we found that the pathogen was circulating the most in that 5 to 15-year age group. So I guess my own contribution to this sphere of tools, I am mostly a user of these tools, but this is my first foray into developing a tool like this, is the AMR spread R package that I hope to release later in the year. So what this software does is it takes a dated or undated tree. It takes AMR determinant data you've screened using another software tool. Maybe it's been ResFinder, maybe it's SRST2, maybe it's any of the common tools that we like to use for doing this analysis. And it takes sample metadata in the form of country or region and year of isolation. And what it produces are visualizations and line lists of AMR dynamics. So principally what this can tell you about is the frequency of AMR emergence in a given setting or across settings compared to local transmission and international importation of AMR among your pathogen group of interest. It can help you where you've got dated trees to look at the emergence dates of resistance when this has occurred. It can look at the dates of local transmission, dates of importation and dates of exportation as well. It can help you track different resistance AMR lineages over time. It can help you look at different international spread patterns as well of resistant strains. So I do hope to release this later in the year. So we've looked at what we can consider to be phylodynamics, what are some of the key concepts are. We've looked at a very brief overview, I do realize, of phylodynamic approaches and the steps involved in these analyses. We've looked at some of the things we can do once we've got a dated tree that might help us tackle different parts of the pathogen epidemiology. And I'd just like to close this webinar on a case study looking at the emergence and spread of multi-drug resistant typhoid in sub-Saharan Africa. So for those who might not know much about typhoid, it's caused by an organism known as Salmonella typhi. This is the bacterial agent of typhoid or enteric fever. And each year worldwide, we see around 10 million illnesses that result in more than 100,000 mortalities. And the control of typhoid is complicated by antimicrobial resistance. So when this study was published in 2018, at that point in time, there was limited data on the different genotypes of Salmonella typhi that were driving typhoid illnesses. So the Typhoid Surveillance Across Africa Project Consortium sequenced 249 Salmonella typhi that were isolated between 2008 and 2015 from 11 different countries in the region, and they were subjected to Illumina whole genome sequencing. So the first stages in this analysis was to carry out pathogen genotyping using the genotypy framework that classifies different typhi sequences into different strain types or genotypes using a phylogenetically informative nomenclature. And then the whole genome sequencing data we used to infer a core genome and maximum likelihood phylogenetic tree that included around 2,000 global context genomes. About 500 of those were from sub-Saharan Africa, giving a total of 2,306 samples. And what they found through these two analyses is that there were 12 different genotypes circulating through the countries that were studied. And of these, 90% belonged to 3 different genotypes. 40% belonged to H58, genotype 431. This is one that's been really well studied in the typhoid field. It's referred to both as 431 and H58 on the basis of the current and previous typing schemes for the pathogen, with the H58 representing haplotype 58. The next most common genotype circulating throughout these settings was genotype 311, which was present at 39%, and genotype 232 was present at just 12%. So what was really interesting in this study was that these different genotypes of Salmonella typhi were geographically restricted. We found that H58 was restricted to East African countries, and the genotypes 311 and 232 were restricted to Western African countries. When they looked at multi-drug resistance and plasmid content among these samples, what they found is that 2 genotypes, genotype 311 and 431, were both multi-drug resistant. They had acquired genes conferring resistance to chloramphenicol, cotrimoxazole, and ampicillin. And this had arisen in different parts of the tree, in different genotypes, meaning that this multi-drug resistance phenotype or genotype had arisen on multiple occasions in the pathogen populations in these settings. And what they found is that both of these genotypes had acquired multi-drug resistance through INCH1 plasmids, but they were different lineages or different variants of the plasmid. And you can see this reflected in the different colors shown on this final color bar of the heat map here. Similarly, you can see that while they both are multi-drug resistant, they both have different combinations of genes that ultimately give that multi-drug resistant phenotype that you see here. And both of those plasmid variants carry different combinations of antimicrobial resistance genes. So seeing this in genotype 431 or H58 wasn't too surprising. This is well known to be associated with drug resistance and intercontinental transmission. And we were aware prior to this study that this was circulating in sub-Saharan Africa. What was more interesting and less known about was multi-drug resistant 311 in this particular setting. So we chose to carry out a phylodynamic analysis of genotype 311 to better understand the emergence and spread of this particular pathogen genotype. So for this, root-to tip regression analysis was carried out using a core genome maximum likelihood phylogenetic tree. And based on the regression analysis that you can see here, this appeared to have clock-like behavior. We sort of had a weak temporal signal that we thought we could see in these data. And on the basis of that, we chose to go ahead and progress our analysis to the model testing stage. And the best fitting model that we found was a relapsed clock model. You might also see this referred to as an uncorrelated clock model. And we also used an exponential growth coalescent tree prior. And we've sectored these through an informal model testing approach. And what you can see here from the second plot that I've nested next to this phylogenetic tree here, this phylodynamic tree, is that our date randomization testing did indicate a robust temporal signal among these data. And I just wanted to give a shout out to Sebastian Duchenne, who's currently a lab head at Institut Pasteur, who helped a lot with this analysis and really helped set us on our way for this. So what we also did with this analysis is we used discrete trait mapping to better understand the spatiotemporal dynamics of this genotype. So we modeled at the tips of our tree geographic location where all of these different samples had come from to better understand what we could learn about the origin and spread of this pathogen using discrete trait mapping or phylogeography approaches. So what this revealed to us is that Ghana was the most likely origin of this pathogen population of this genotype. We believed it probably, from the data we had at the time, emerged then. And we've seen that subsequently spread to other countries that are neighbouring Ghana. For example, we can see some teal-coloured tips in that tree representing spread into Burkina Faso. And when we look at the purple branches and tips in that tree, what we can see is that we have descendants and multiple introductions that have occurred into Nigeria. And when we look at this a bit more closely, what we can see is that one of these introductions into Nigeria has led to this local expansion of this pathogen population, this MDR311, and this occurred around 2010 to 2011. And when we look at TyphoNet, which we developed recently as an online dashboard for typhoid genomic surveillance that gives you estimates and prevalences of AMR and pathogen genotypes over time and geography, that genotype 311, shown in sort of a blue color on this particular diagram, is still the most prevalent genotype in Nigeria to date. So I hope what you can see that we've learned from this is that by combining multiple types of data and methods, we combined metadata in the form of year and location of isolation, pathogen genotype dating, AMR determinant and plasmid screening, maximum likelihood trees, and Bayesian phylodynamic inference, we've been able to identify that there's a problematic strain of multi-drug resistant Salmonella typhi circulating in West Africa that is different to H58. We've identified that there's regional spread occurring of this genotype that emerged in Ghana before spreading internationally on several occasions, driving that expansion event that occurred in Nigeria in the 2010s. So if you'd like to find out more about this, I do encourage you to take a look at this paper that we put together by our Park and colleagues in 2018 in Nature Communications. So thank you very much for your attention. I realize this is a very brief overview and whirlwind tour of phylodynamics. We've covered some basics around what phylodynamics is and how you might like to use it, a very brief overview of the analytical process, what you can do extra once you have your data trees that can help you better understand the genomic epidemiology of a pathogen. And we've shown how this can all be put together in a case study of MDR typhoid in sub-Saharan Africa. If you're looking to learn more about any of the topics covered today, this is intended more to be sort of an introduction to the field rather than a comprehensive cover or webinar. What I would suggest to you is there's an excellent book that I've indicated in the top left of this slide here. This has been a really helpful tool for me in my learning and progressing my knowledge in this space. I would also recommend the Beast and Beast 2 websites. They have excellent tutorials that can walk you through doing some of these different analyses. A lot of them will be based on viruses, but a lot of that knowledge is transferable if you look at how you might refine those and published examples in the literature. I would also highlight that Taming the Beast is an excellent source of tutorials for learning these techniques as well. They do run a course each year. I believe the next one's in Beijing, and that runs every year. So if registration has closed for this or any of the other courses I've highlighted here, they will run again, most of them. And I've highlighted here SysMid, which is a course that runs every year in the States as well. This is also available online if travel's not an option for you right now. And several other workshops that are either coming or do run regularly that might be helpful to you. So I hope you found this overview to be helpful to you. Thank you again for having me here to speak, and I'd be happy to take any questions that you might have. Thank you very much.
Thank you so much, Dr. Zoe Dyson, for that very informative and actually very clear breakdown of doing phylodynamics analysis and also the caveats while doing the analysis. We do have a couple of questions. I'm going to try and group because there are some of them that actually wrote together. So firstly, I'm going to group these together. So can you, is there a minimum date range needed for this analysis? I'm going to group three together. Can temporal trees be informative for outbreaks that span or that's less than 6 months, span less than 6 months? And what is the minimum sample size needed to conduct such an analysis?
Oh
So
gosh,
maybe
so I feel like this is not going to be a popular answer, but it is an honest one. And the answer is pretty much for all of those, it depends. So in terms of your minimum sample size, I'd say the more critical thing is probably temporal span. Do you have enough temporal span based on how quickly your pathogen evolves that you can capture that temporal signal that means that you can then go ahead and do that analysis? So some pathogens evolve more quickly than others. I do a lot of work on typhoid that acquires one SNP every 2 years, maybe one every year if that's in the core genome. So, what I would use for those analyses might be different to what you can use for yours. So it really does depend on how rapidly the pathogen evolves, the temporal span that you have in your data. It's quite difficult to give a ballpark figure. I'm sorry about that. I would highlight that there's a variety of data sets that have been analysed in that publication by Duchenne and colleagues in 2016. Looking at some of those data might be informative for you as well if you're working with bacterial data. I hope that helps.
Thank you. And then the first question that was actually asked in that series was, you mentioned that this may not work, or it may not be as clear if you're using virus genomes. Could you elaborate on that a bit more?
Yeah, sure. So again, it depends on the pathogen you're working at, on the data set that you're working on. But sometimes for some viruses, your regressions might be much more. You might have smaller residuals. You might be able to. They might be less dispersed than you would see when you're working with bacteria. And so there's a variety of different things that can influence that. In terms of the analytical processes and the models that you might select for a viral population, I'm not a virologist. I've never worked with viral data. I can't really elaborate too much on that, unfortunately. But I would redirect you to some of the tutorials that are on the Beast 1 and Beast 2 and Taming the Beast websites. They might be helpful to you. But also exploring the literature of teams that might have tackled these problems before and getting a feel for what they've done and what baseline looks like in the community and what consensus looks like in the community. I do hope that helps.
And just to mention again, this is exactly why we're saying if we could get like people who may not even be experts in the field in these phylogenetic working groups, these are the type of questions, these are the types of variety of pathogens that we need to consider when we are building and developing that guidance document. So it's very important that we have you at those meetings so that we actually build a product that is a good overview of or actually meets most people's needs. Maybe this one is a short one. I know you mentioned that you used GG3 somewhere. Was that also used for your figures in the Typhy paper?
I don't believe we used it in the paper that I showed you. We have used it in studies of typhoid in Nepal. There's a paper from Brito and myself as the lead authors in that from 2018 in PLOS neglected tropical diseases you could look at. There's another one that we published, I believe, in 2022 in PLOS neglected tropical diseases looking at typhoid in Papua New Guinea. If you're looking for examples of some figures you can generate using GG-tree, they're the ones that come to mind. But I do recommend it to you. The learning curve, if you're not familiar with R, might be a little steeper, but you can make some really nice and really flexible diagrams using that. Another option for you might be Figtree. There are some customizations that you can do if you're more comfortable with a graphical user interface. That might also be a good one to consider. I hope that helps.
Thanks, Zoe. Maybe one last one. We're on the hour, but if I can squeeze this one in.
Of course, please. My pleasure.
I know this is for those who are, fortunately, some of us have had some background on phylodynamics, so we could follow along. But how do we actually, since we're dealing with people who are working in public health, is there a way in which we can communicate this phylodynamic findings to public health decision makers? That I don't know if there's a specific way.
So I think there's been some good examples of using phylodynamics, particularly surrounding COVID. I think those are the examples to look towards in terms of how you might consider communicating these data. And they are really useful data to public health people who can then and those who are in a position to make decisions based on the data. So I think definitely looking towards how that data was integrated in the COVID pandemic will be the way to move forward with that.
Okay. Then thank you so much, Dr. Zoe Dyson. Thank you for everybody who joined. Thank you to the PHA4GE group who availed the slot for Dr. Zoe Dyson. We hope to see all of you signing up, and we hope to see you in the pipelines and visualization meeting. And Dr. Zoe, also you as well. Hopefully in the future, we can invite you back to give us some updates on some of the work that you've done, and hopefully by then your tool will be published. So thank you very much, though, for all of the effort and the time that you've already given us. I hope you have a good holiday when you eventually have it.
Thank you so much. It's been a pleasure, and thank you so much to everyone for attending.
Thank you. Thanks, everyone. Have a good rest of the evening. Bye-bye.
Bye.